both CD4 and CD8. CD4 are T helper cells and CD8s are cytotoxic T cells, right? This area that you see, so this basically, these are two lobes of thymus, 1, 2. This is heart. It's actually sitting on top of the heart, right? And present in the thoracic cavity, right? When you are a kid, so it's larger in size, and when you grow older, so there is thymic atrophy, shrinkage of thymus. All right. So there are some cells which actually are responsible for maintaining lymphocytes. Okay, those cells actually are called nurse cells. They secrete this molecule that we call as lymphopoietin. It's a growth hormone for lymphocytes. It facilitates the survival of lymphocytes. Okay, so you find lymphopoietin being secreted in thymus. The other thing that we also talked about was that thymus is the site where, if you actually want to study apoptosis, so you can collect thymus and you can actually find out a lot of cells are undergoing apoptosis. Okay, so the cells which actually start their journey from double negative to double positive to single positive. Now I know these are actually these will look like a jargon at this time point, but we'll talk about the differentiation of these cells. T cells actually, as I said, they can be of CD4 or CD8 lineage. Now, during the development of these cells, so you have a stage where your T cells express both of these molecules, CD4 as well as CD8. Okay. And even a preceding stage that you have in thymus, so you don't find any of these molecules expressed by your T cells, right? Okay, so you start with double negative, but they do have some markers which are associated with T cells, and that is why we call them as T cells. Okay, CD3 is one of those. After double negative stage, they become double positive, expressing both CD4 and CD8. And then if they come in contact with MSC class 1, which present this peptide, which is not autoreactive, so these cells will become CD8. And if these rather double positive cells come in contact with MSC class 2 derived peptides, they become CD4. So we'll talk a little bit more about the development of T cells in thymus. But just for the time being, remember that thymus is the site where T cells development take place. Pretty well organized tissue. More or less the structure is similar. So you have this capsule, trabecula, and then cortical and medullary region, where you have this T cell development process taking place. Okay, so they are sensed of in a way that if you find autoreactive cells, so that means if these cells react to peptides which are of your own origin, these cells die. And if they don't, they survive. And that is how you maintain that autoreactive T cells are eliminated and your cells which do not react to your own antigens, they are selected and eventually come into the peripheral lymphoid organs. Okay. So we in previous slide we talked about actually the presence of this thymus, right? And now there was actually a paper which showed, this was actually in 2006. They provided evidence in mice for a functional second thymus. Okay, this is actually surprising if you think about it, because thymus, for ages, we have been knowing that there is one thymus which is present in thoracic cavity on top of your heart, right? And is the site for development of your T cells. In this study, they actually they wrote this paper in Science, showing that there exists another thymus, which is actually smaller in size, much smaller than the thymus that you have. It's like the size of a lymph node. Okay, this is also called a cervical thymus because this is present in the neck of these mice. Okay, typical structure of medulla and cortex were found in these second thymi or thymuses, whatever you want to call them, right? And it actually supported the development of T cells, right? Now, why this observation was relevant? So for a long time, when people were studying the role of T cells in different diseases, what they used to do was they remove this thymus because it's quite well evident a tissue. So it's easy to remove thymus by a process that we call as thymectomy. So you can actually remove this thymus out of these animals and then try to study what is the role of T cells in disease progression or control, right? Now, in many of these studies, what they showed was despite removal of this thoracic thymus, you still find some T cells. And they were never able to provide an explanation where these T cells came from. And when they actually provided evidence to demonstrate that second thymus is present and is functional and is able to support development of T cells, so this somehow provided explanation to those observations. Okay, so if you want to read more about this paper, you can read it. This is the reference, 2006 Science. All right. How come attendance is so thin today? Only 59 students out of 90. Any explanation? So Vishnu, you have an idea?
No, sir. I mean, there's no specific reason. I don't know. Like,
Because many of you are in hostels and internet seems to be working, right? Well, anyways, okay, so the other organ that we call actually as tertiary lymphoid organs. And these are called lymphatic nodules. Okay, so they are oval shaped structures. Okay, basically a collection of these lymphocytes and surrounded by some extracellular matrix. It's not surrounded by connective tissue. This is not highly organized. It's just aggregation of these lymphocytes, and these predominantly appear once you are infected with one microbe or the other, particularly, for example, after influenza virus infection. So you tend to develop these tertiary lymphoid organs. Okay. that we call as, for example, mucosal associated lymphatic tissues. So there are different names. You have organized NALT, nasal associated lymphatic tissues. Okay. MALT is mucosal associated lymphatic tissues. But they are not as structured as you have for your secondary and primary lymphoid organs. Another example is Pius patches, which are actually present in your small intestine and are very critical for inducing responses in your intestinal tract. Okay. Pius patches. Okay. Now, for example, if you cut open a mouse, sacrificed mouse, so you can enumerate a lot of these lymphoid organs. Okay. Primary and secondary lymphoid organ. You know, bone marrow is one of those primary lymphoid organs. The second is thymus, right? Which actually is present here, right? And then you have, if you read through this list, there are so many different types of these lymph nodes: superficial cervical, deep cervical, mediastinal, axillary, brachial. Then you also have the spleen, right? Pyloric lymph node, renal lymph node, mesenteric lymph node present in basically the mesentery is, what is mesentery? In intestine, the connecting portion, basically, so you have different fold of this intestine, and these are connected through this connective tissue. Okay, so this is called mesentery. So, and you find actually in bunches these lymph nodes, these we call as mesenteric lymph node. You find a lot of cells, a lot of lymphocytes are present there, and very critical for providing defense to your gut or gut.
Excuse me, sir.
Yeah.
Sir, is lymph nodes and Peyer's patches a part of secondary lymphoid organs or tertiary?
Well, you don't call Pius patches as secondary lymphoid organs. Okay. You tend to call them as tertiary lymphoid organs. Okay.
And sir, what is the basis of their classification?
Classification basis is how organized is the structure. Okay, whether you have these cortex, medulla, capsule, trabeculae, well organized germinal center. They are surrounded by connective tissue. So all these things will basically classify these cells as organized or not so well organized, or just a mere aggregation of these cells.
Right, sir. Thank you.
Yeah. Okay, you can go through this slide. So you can actually name these lymph node as different types of lymph node. And this comes handy when you actually start doing your immunology research. Because, for example, many a time you have to do foot pad infection in a mouse to investigate immune responses. Okay. Now, if you do a foot pad infection, so the response is going to be generated in those lymph node which are actually draining to this site. We call them as popliteal lymph node. If you are, for example, doing an ocular infection, your draining lymph node are going to be these cervical and these superficial cervical lymph nodes, right? And if you are, for example, doing a systemic infection intraperitoneally or intravenously, so the response can be measured in spleen. Okay, so that actually becomes important depending on what route of infection you are you're using for your investigation. And obviously, so for practical purposes, for example, if you for example have some infection in your tooth, right? So the lymph node which are actually draining to their site gets swollen. So that means you actually are starting to generate an immune response against whatever has infected your teeth, right? So this is how you classify them as draining versus non-draining. Non-draining would be those lymph nodes which are far away from that place. Okay. And why we call them as draining versus non-draining? Because in the acute phase of the response, the major response you actually can measure is from the area, from the lymph node which drains to that tissue, and not in an area where this is actually far away from that site. However, after some time, some of these cells also tend to migrate to other locations, and this is called a diaspora effect. Okay, so don't worry too much about all these things as of now. This is just to give you a picture that there are so many different types of lymphoid organs you can find even in mouse, which actually is used for investigating your immune responses, right? Now, how this is actually a slide which shows you how you can visualize the concept of drainage. Okay, so in this case, what they did was they injected these dyes, okay, which fluoresce at different wavelengths. Okay, start 605, 655, 800s, 565, 705. Okay, different wavelengths of dyes which actually fluoresce at different wavelengths. Now, if you, for example, inject this dye in one of the 4 limbs, so, and very early after injection, within a few minutes after injection, if you visualize, so these 2 lymph nodes will lighten up. Okay, as you can see here. If you use another dye, for example, you inject this in ear vein or in ear, right? So these other 2 lymph node will lighten up. Okay, and then if you inject basically under in the neck region, so then you lighten up these ones. So this is to suggest that wherever you inject this dye, and this dye can be likened to the antigen that you have injected in that area, which is fluorescing in nature, and that's why you are able to visualize the structure of those or the localization of those lymph node, and you can find out that the draining lymph node for that area are the one which are highlighted. Okay, all right, so you can actually generate pretty fancy pictures when you do such kind of experiments, right? To visualize and that these are the draining lymph node for neck, these are for forelimb, and for ear, for the other side, and so on and so forth. Anyways, now if we talk about in general the development of your T and B cells, right? So you have these bone marrow, which are actually the site of hematopoiesis, right? So you start from hematopoietic stem cells. These hematopoietic stem cells then become, rather than differentiate into common lymphoid progenitors, right? And if these these common lymphoid progenitors have to further differentiate into B cells, so they stay back in bone marrow, complete their journey of positive and negative selection, and once this is made sure that you have selected only those B cells which are not reactive to your own antigens, but they would recognize foreign antigens. So those cells will exit bone marrow cells, and then they will be present in these peripheral lymphoid organs, okay, spleen and lymph nodes. This is for B cells. That means the development process of B cells actually take place in bone marrow. Now, these hematopoietic stem cells, again, these are basically lymphoid progenitor cells. And if they have to give rise to T cells, they don't develop, or they don't actually complete their journey in bone marrow. So they leave bone marrow, get into the thymus. Okay, thymus is the organ that we talked about earlier on. Okay, so here in thymus, so they actually start their process of positive and negative selection. And if you make sure that you have not selected autoreactive cells, so those cells then populate peripheral lymphoid organs. Okay, now once these cells are present in these lymphoid organs, now you can start generating your B cell mediated responses or T cell mediated responses in response to either microbial infections, injected antigens, vaccines, developing cancer in your system, and in some cases, autoimmune diseases as well. Any question on this slide?
Sir, what did you mean by positive and negative selection?
We'll talk about those. But positive selection apparently means if you're developing T cells, somehow do not make a contact with MSC class 1 or class 2, so they die. Okay, so this is a positive signal. So if they make contact with MSC class 1 and MSC class 2, so they survive. Okay, now negative selection is if they make contact with MSC class 1 through this peptide, and this peptide is not autoreactive. So these cells are selected. If this peptide seems to be autoreactive, so they die. This is called negative selection. So these processes are very finely dictated. Okay, so don't worry. So we are going to talk about some of these processes in subsequent classes. But essentially, this is what positive and negative selection means. And this is to ensure that you do not develop autoimmune diseases because of your inefficient selection system in your lymphocytes for T cells and for B cells both. Okay, this is not to say that this is 100% foolproof, because some people tend to develop autoimmune diseases, diabetes, rheumatoid arthritis, myasthenia gravis, multiple sclerosis. These are all the examples of your autoreactive cells, autoreactive autoimmune diseases, and contributed by these autoreactive T cells or B cells. Okay, but efforts are made to ensure that you don't get autoreactive cells coming into the periphery. Even if you get some cells coming into periphery, so there are mechanisms to suppress those cells. Yes?
Yes, sir.
Okay, so yeah, again, so this actually is this slide actually shows you how you actually can detect these blood cells. Okay, although this is electron microscope, and where these cells are stained with different markers. Okay, for example, platelets were stained with in purples. Okay, so the purple. T lymphocytes were stained in green, monocytes in gold. Okay, so you can actually use different dyes to stain these cells, and this is actually a very fancy technique. But this came into being much later. So earlier, so how people used to detect these different cells in your blood was by a simple staining. Okay, so almost 50, 60, even 70, or rather even I would say 100 years back, when these cells were being um, started to being discovered. So these were the simple techniques people used to use. Okay, so you take out some blood, you make sections, and you use these dyes. And one of these stain that you can use is a right stain. Okay, this stain, and then you count the number of those cells. For example, in different fields in a slide, if you count 100 cells, out of 100, how many of these cells are of this particular type? Okay, which we call them as neutrophils. As you can see, the shape of the nucleus. Okay, these are called polymorphonuclear cells. You have highly lobulated nucleus in neutrophils, right? And the staining characteristics of nucleus and cytoplasm are also different. Okay, so this will tell you that if you get something like as this, and although this is a very old technique people developed way back in 90s or maybe 1920s or around that time. Okay, but still in clinics, if we, for example, want to get your DLC is differential leukocyte count, DLC analyze. So these are the kind of techniques these lab technicians use. Okay, to detect whether you have neutrophilia, whether you have more monocytes, whether you have more lymphocytes, or whether you have allergic responses because you find more number of eosinophils in your blood. Right, monocytes actually are where you have this kidney-shaped nucleus. Right, lymphocytes are basically where almost the whole area of this cell, this lymphocyte, is covered with this nucleus. Okay, roundish nucleus. Eosinophils, they are not highly lobulated, but you find these two lobes at least in eosinophils. Now, depending on what stain do they take, so you can classify them as eosinophils or basophils. Okay, depending on the characteristics of cytoplasm as well as nuclear staining. Okay, so we are going to talk a little bit more about these cells, and we already have talked about this slide in the very beginning. But just to remind you that you start with stem cells. They differentiate into lymphoid stem cells, myeloid stem cells. Lymphoid stem cells give rise to B, T, and NK cells. B cells, upon recognizing antigens, become activated. They make this Y-shaped structure that we call as antibodies. Some of these B cells become memory B cells. T cells, they get activated by seeing these peptides, not the whole antigen, and the whole antigen needs to be processed through this machinery that we call as proteasome, and it can be still fine-tuned. They are phagolysosome and other kind of machinery, right? But predominantly, so these whole antigens are processed to generate peptides, and those peptides, if loaded onto MSC class 1, so going to activate your TC cells or cytotoxic T cells or CD8 positive cells. And if these are loaded onto your MSC class 2 molecules, they are going to stimulate your T helper cells. These cells will become effector cells. They will start making all kind of cytokines, granzymes, perforins to perform their effector functions. And a pool of these cells, which rarely account for 5 to 10%, will become memory cells. Okay, these are your lymphoid stem cells. Myeloid, all these cells that we discussed in previous slide, neutrophils, eosinophils, basophils, mast cells, monocytes, they are part of your myeloid stem cells. Okay. These cells, just a second, of your immune system originate from precursor cells in bone marrow, right? And the process is called hematopoiesis, wherein your blood cells develop. Okay. You can read this other line. All the cellular elements of blood, including the cells of your immune system, arise from pluripotent hematopoietic stem cells in bone marrow, and then they further differentiate into one lineage or the other, giving rise to either myeloid cells or lymphoid cells. Okay. This slide actually summarizes everything in this whole process. You have these pluripotent stem cells, which will give rise. This actually is, these are present in bone marrow. And if these cells have to give rise to lymphocytes, the first step would be common lymphoid progenitors, which will give rise to B, T, and NK cells. Once they pass through this process of selection criteria, they get into lymph node, where they get activated, become effector cells, carry out their protective mechanisms. Myeloid cells will give rise to these granulocytes or platelets, right? Erythrocytes. And in blood, so you call them as, depending on what morphology do they exhibit, what staining characteristics do they acquire, what markers do they express on their surface, right? Neutrophils, eosinophils, basophils, monocytes. So all these cells are coming from your myeloid stem cells. Okay. One more cell actually, which is very critical, is we did have discussed a little bit about this, is genetic cells. Initially, these are immature, but once these immature cells, they see or they sense the presence of these antigens, they become mature dendritic cells. And by maturation, I mean they start alteration in their morphology. Okay, so the morphology is altered, these projections become bigger, they start upregulating some of these chemokine receptors, they start making some initial set of cytokines. Okay, so this is called maturation. They can now efficiently take up antigens, and once the antigens are taken up by these dendritic cells, they are going to engage your T cells for the initiation of your adaptive immune responses. Okay. Now, one more thing that you need to remember is these monocytes are present in blood, but when these monocytes from blood gets into tissues, we start calling them as differently. Okay, you start calling them as mast cells or the macrophages, and their name actually varies depending on which organ you are looking at these cells. Okay, that we'll talk about in some of these slides early, that we are going to talk about. Okay, they are actually coming from these monocytes in the beginning. Okay, all right, so we already have talked about it, so I don't know if we need to go through it again. Same thing, common lymphoid progenitors give rise to B, T, NK cells, get into lymph node, activated by seeing these antigens. How these cells see antigens is through different mechanisms, different ways, different pathways are engaged, and common myeloid cells will give rise to all these cells types we talked about, right? Now, I think this slide is also a little important because this will give you some idea so how things happen. Myeloid lineage comprise of most of the cells of your innate immune system, right? And your lymphoid lineage will give rise to your lymphocytes and NK cells. Lymphocytes are part of your adaptive immune system, and NK cells are part of your innate immune system. But this definition is also being revised in recent times. But still, if you want to call them as part of your innate immune system, so you're all right. Okay. Just to make you more educated on why we call them as part of both adaptive and innate immune system. So I'll give you a citation. Okay, so it was actually paper was published in 2009 in Nature. Okay, so again, so you have these lymphocytes, which actually are, so if you count, for example, 100 cells, out of 100 cells, so lymphocytes are usually from 25 to 35%. Monocytes are close to 4 to 6%. Eosinophils are 1 to 3%, and basophils, sometimes you may not find out of 100, but their range actually varies to 1%. Neutrophils are the predominant cell type you will find on a piece of slide that you have stained with these dyes. Okay, you cannot miss out neutrophils. Okay, so lymphocytes and monocytes, you also call them as granulocytes, and rather, well, these are actually agranulocytes, and these are granulocytes. Okay, so this is wrong. Correct it once I upload. All right, so these are the cells and their relative proportion. Now, for example, if you get your blood tested, and if someone told you that your lymphocyte level actually is 50% in DLC, so you can immediately sense that either you are running a viral infection in your system, or you are developing probably some kind of cancer. Okay. If you, for example, find that your neutrophils are close to 80%, so you can actually see, or you can actually assess that there could be a chance that you are running a bacterial infection. And if you have more number of eosinophils, probably you are allergic to something or the other. Okay, so these are the indications that you get from DLC data on your blood. Right. Okay, now very briefly, we are going to talk about the function of each one of those cell types that we have talked about. Macrophages, they are predominantly responsible for phagocytosis and activation of bactericidal mechanisms. Okay, so they produce a lot of the reactive oxygen species, reactive nitrogen species, and are responsible for killing the engulfed microbe intercellularly, right? They're also very critical in antigen presentation. Okay, so they take up these antigens, they chew them into small pieces, and they help present to T cells. So that's their major function, macrophages. As the name suggests, macro means big, phages means the cells which eat up stuff, right? Dendritic cells, major role actually is in antigen presentation. They are actually called as professional antigen presenting cells. Their role actually is even more critical than macrophages in antigen presentation. They can very efficiently take up antigens from peripheral sites because of the projections they have, and they can actually scan through a lot of area, a large area, right? Neutrophils, major role, phagocytosis and bactericidal mechanisms. Okay, so these are the two things they carry out. Although, so in recent times, we are also discovering that they can actually release these, for example, once they are, we call neutrophil as sloppy feeders, okay, in slang language. So when they eat up these microbes, okay, through phagocytosis, they don't do it very efficiently. So they release a lot of stuff. Okay, so a lot of these small dots that are actually shown here. They are actually granules which carry a lot of these enzymes, proteolytic enzymes, and many more dangerous stuff, which actually is responsible for killing these microbes, but they are also released outside these cells once they take them up. Okay, and so what that means is, so the clearance of a microbe by neutrophil is not a very efficient process, and it causes a lot of inflammation. Okay, so if you want to kill off a microbe, so you don't want to engage neutrophils to the extent that you end up developing all kind of inflammatory diseases. Okay, so you have to control their function as well. The other thing that I wanted to mention was, if they release their DNA, nucleic acid, and the released nucleic acid can also engulf or can also trap this extracellular bacterium, which actually is present in the close vicinity. Okay, so these are called NET. Okay, neutrophil E, I forget about what E was, but I'll tell you what. That's when I remember. Okay, so these basically are trap mechanisms for engulfing these microbes if the DNA is released from these neutrophils. Okay, basophils. Basophils, their function for a long time, it has been unknown. They have been described to be playing a role in some allergic reactions, but still not well established. But in a report which was published again in Nature Immunology in 2009, they showed that basophils actually can serve as a good antigen presenting cells, although their number is too low. Okay, so you don't really call them as professional antigen presenting cells as you have for macrophages, dendritic cells, B cells. But if you are able to isolate these cells from a host, you can demonstrate that they are actually also very crucial in antigen presentation. Now, what they did show here was, so one of the classical thing that APC, any cell which actually performing as APC, antigen presenting cells, they have to express a lot of MSC class 2, right? And this is what is shown here. These are basically the those cells, right? They express a lot of MSC class 2 on the surface. And when you give this antigen, so they even start upregulating more, right? So they actually express, they are not expressing this this MSC class 2 constitutively, but once they take up antigens, they tend to express MSC class 2, right? Now, then they showed that if you engage these APCs, okay, so rather these basophils with specific TCR expressing cells, for example, papain specific T cells. So these cells, these basophils were able to form immunological synapse with T cells, as you can clearly see. So if you stain for TCR, the maximum staining of this TCR actually is towards the side of this basophil. And this process is called cupping, okay? Formation of immunological synapse, that all the molecules which are responsible for making these contacts between APC and your T cells. Okay, so let's say you have this T cell, you have your APC. So this is going to basically have this MSC class 1, class 2 molecules presenting this peptide. And on the other side, you have these T cells. Normally on T cells, on naive cells, everything is distributed equally. Okay, but once you have this engagement, so all these molecules tend to migrate towards one side. And this is actually what is shown here in form of the staining of this TCR. Okay, so all these TCRs get recruited towards the synapse. Okay, so this actually shows that basophils can serve as antigen presenting cells. Okay, 1048. So if you want, I can stop or I can continue for maybe 2-3 more slides. Mast cells, you know that they are actually very good in releasing these histamines, right? Responsible for allergic responses, right? And finally, you have your NK cells. So I think this will take some time to explain. So I don't want to talk about NK cells today. So, however, if you have any question up to this point, so let's talk about those in next 4 or 5 minutes.
Excuse me, sir.
Yeah.
Sir, is there any particular reason why we can see such a diverse morphology in terms of the nucleus of these cells?
Well, not that I'm aware of. Well, there has to be, because nothing happens in a system without a region, right? It's just that I'm not aware of. So what could be the region? I don't know. So maybe because the major function of, for example, neutrophils is to kill stuff, right? So what the phagocytose, and maybe in so doing, if they also tend to chew up some pieces of its, I don't know, own internal structures. So I don't know, the shape of nucleus might change. Well, this is just a I don't know, hypothesis or maybe a random guess, but I don't really know. Okay, so maybe you could find out.
Okay, sir.
So
Yeah.
So initially, you had told that these monocytes give rise to macrophages and dendritic cells, but in one of your recent slides, it's mentioned that other way, like monocytes are giving rise to macrophages and basophils. Like, could you go back to previous slide, like
Which one? This one?
4 slides back, like
Here.
yes, sir. Yeah, mast cells and macrophages.
Yeah, well, mast cells they are all part of your innate immune system, right? Now, if these monocytes actually are present, monocytes, see, more or less these are these are having the same lineage, and almost conserved molecules expressed by these cells, either on surface or intercellularly. However, depending on their location, from blood, if they get somewhere in a particular tissue, okay, so then you start calling them as macrophages. Okay, and let me also tell you that many people, many scientists in field still believe that dendritic cells are actually macrophages, a different type of macrophages. Okay, It's just that they have modified their structure in a way. They have started generating a lot of these projections um, so as to surveillance of antigens in a longer area, and they can engage efficiently your T cells. And this is just a modified function of your macrophages. Okay. And one of the proof for that actually is that CD11B, which is also called as MAC1, it's an integrin, which is expressed by macrophages, is also expressed in large abundance on your dendritic cells. Okay. And that is why, see, these cells were actually recognized for a long time, almost around the time of Eli Mechnikov in 1930, okay, or even way before that. But very recently, in 2011, actually, this field, the dendritic cell, actually were recognized as a separate lineage. And this Rolf Steinman actually who showed that these are actually a separate class of these cells and very critical for engaging your innate and adaptive immune responses, right? And he was given Nobel Prize for that. But again, for example, you can actually generate dendritic cells from macrophages or from bone marrow derived bone marrow cells just by adding GM-CSF and IL-4, two cytokines. If you add to your bone marrow cells, you can generate dendritic cells pretty easily, right? And then one more molecule they start expressing is CD11C. 11B remains same, but CD11C is upregulated. So you don't get confused that which cell is giving rise to what. Okay, so there is always some level of plasticity, which can actually decide their fate and function, right? And these are just the different names people have given. A lot of people, as I said, in the field still believe that dendritic cells actually are macrophages. They are modified macrophages. Okay.
Fine, sir. And sir, one more short question that we know that if T cells is not expressing CD4 and CD8, then there can be two reasons. One is it is in the double negative stage, or otherwise it can be like the development is not complete, like it's not proper. So how do we differentiate both of these cells if they are residing in the same location?
Well, see, if the differentiation is not complete, so that means, or even if the differentiation is complete, these cells are removed through apoptosis. Okay, so you don't detect them. Second, if they don't express any of these molecules, yes, they are actually considered as the primitive cells, which will start their process of development. Okay. And you find actually a major chunk of these cells in thymus which are not expressing either CD4 or CD8. Okay, we call them as double negative. This is not to say that you will always find these cells in thymus. In some disease conditions, some of these double negative cells can also appear in periphery, but that's a disease condition. That's not normal physiology. Okay. Now you're trying to ask probably that whether these, so how do you call them then as T cells, right? No. So
No, sir. I mean, both of the T cells are like, if the differentiation is not complete and like it is in a double negative stage, like how do we differentiate them? Or even how does the body differentiate them that these cells should undergo apoptosis and this is in the double negative stage?
well, so your body has mechanism to give chance to every single developing cells making its way to the periphery. And how this is happening, so we are going to talk about in the part when we talk about T cell differentiation. Okay. But what I said today was that the journey actually starts from double negative population. And you can find these double negative cells also in bone marrow. And bone marrow is not the site where T cell development process is completed. Okay. So these double negative cells actually, they make their way to thymus. And there, depending on the environment, the lymphopoietin that you have, the engagement of MHC class 1 or class 2 you have with the specific TCR expressing cells, or these CD4 or CD8 glycoproteins on the surface of these cells, with MHC not through the peptide but through basically the outside uh, groove in alpha 3 or alpha 2 domain, depending on MSC or MS2. So this, I know this is very complicated at this time point, so probably it will not be very clear unless we talk about those issues. Okay, but for the time being, remember double negative, double positive, and single positive. So this is the process where you have to follow during the development of your T cells.
Fine, sir. Thank you.
Okay, so we'll stop today. So you might have your class at 11, and it's already about to be 11.